CSIR Evolution Chapter 8. 8. Population Genetics. Population genetics is concerned with genetic differences within and across populations, and the dynamics of how populations evolve as a result of the propagation of genetic mutations occurring within the germ lines of individuals. Population can be defined as a group of potentially interbreeding organisms belonging to same species living in a certain area. Members of a population vary from one another. This variation is the raw material on which natural selection operates. Mutation in a gene result in the alteration of DNA nucleotide sequence, producing an alternate sequence, termed an allele. Mutations occur at random, they are not predetermined events. There are several types of mutations, both at the gene level and the chromosome level. If mutations occur at gene level, they result into formation of different alleles and ultimately in evolution. The effect of mutation can be positive, beneficial, neutral or negative, harmful. Chromosomal mutations may occur due to changes in the number of chromosomes inherited, or alterations in number or arrangement of alleles on chromosomes because of deletion, duplication, inversions and translocation. Events. One of the major causes of genetic variations in case of sexually reproducing organisms is the recombination, which involves reallocation of alleles and chromosomes during meiosis. Recombination involves crossing over during meiosis and random segregation of chromosomes to form haploid gametes during meiotic division. A high degree of variations is added at the time of random combination of gametes during fertilization due to independent assortment. There are chances that the newly formed combination of alleles may lead to enhanced reproductive success of the organisms, therefore the entire genome is subject to natural selection. In case of traits which are controlled by multiple genes, i.e., polygenic traits, the right alleles grouped by recombination form the most favorable combination. Variations are not only created, but if they are found to be beneficial for the organisms, they are preserved and also passed to next generation. The sum total of all the alleles in a population is called as the gene pool in context with gene frequency. Small changes in the gene pool accumulate over a relatively short period of time and eventually lead to microevolution. Gene pool composition is not affected by either dominance or sexual reproduction. Evolutionary forces such as genetic drift, mate choice, assortative mating, natural selection, sexual selection, mutation, gene flow, meiotic drive, genetic hitchhiking, population bottleneck, founder effect and inbreeding act on gene pool and changes its composition. 8.1 Assessment of genetic variation within populations. The basic requirement of evolution of a population is the occurrence of variation in genetic material. We cannot observe genetic changes in our everyday life, instead the effect of genetic changes that reflect into certain phenotype, the physical expression of a gene, can be observed. Any given phenotype can be observed in terms of certain character. For example, eye color is a character and brown eye color is the trait or a phenotype. A trait is the specific expression form of a character. In an organism's body, the genetic traits are completely or at least partly determined by the genes. Although the agents of evolution directly act on phenotypes, but for the moment we will concentrate on genetic variation within populations. We will do so because genetic variation is what is passed on to offspring via gametes, eggs and sperm. The unit of macroevolution is population, but smallest unit which can evolve is a gene or even single nucleotide. The genetic constitution that governs a character is called its genotype. A population evolves when individuals with different genotypes survive or reproduce at different rates. Different forms of a gene, called alleles, may exist at a particular locus. A single diploid individual has only 2 of the alleles found in the population to which it belongs, but population as a whole may have multiples alleles, figure 63. The gene pool is the set of all alleles, or genetic information, in any population, usually of a particular species. The gene pool contains the variation that produces the phenotypic characters on which agents of evolution act. There are certain levels of genetic variations for several characters in nearly all populations. Artificial selection on different characters in A. Wild mustard produced many important crop plants worldwide. Plant and animal breeders could achieve such results because the original population had polymorphism for the characters of interest. Laboratory experiments also demonstrate the existence considerable genetic variation in populations. An experiment involved fruit flies, Drosophila melanogaster, with either high or low numbers of bristles on their abdomens as parents for subsequent generations of flies. After reproducing 35 generations, all flies in both the high bristle and low bristle lineages had bristle numbers which were even different and fell well outside the range found in the original population, figure 64. Therefore, it makes clear that there must be some considerable variation in the original fruit fly population on which natural selection can acts to choose out best phenotype. Figure 63, a gene pool is sum total of alleles in a population. The study of evolution on genetic basis is not that simple because genotypes do not uniquely determine phenotypes. For instance, in case of dominance, more than one genotype can give a single phenotype, for example e.g., due to the dominance of allele over allele, AA and AA. Individuals may be phenotypically identical. Likewise, a given genotype may give different phenotypes under the influence of varying environmental conditions. Consider a leaf of a tree or a shrub. All the cells of the leaf are normally genetically identical, still there is a difference in the shape and size of all leaves of the same tree. An oak tree has more deeply lobed leaves which are closer to the top of the tree as compared to the leaves which are in the shade and growing lower down on the same tree. The same differences can be seen between the leaves of individuals growing in sunny and in shady sites. Therefore, it can be stated that although the characters are governed by the genotype, the expressivity is affected by a complex series of developmental processes that are influenced by both the environment and its genes. Figure 64, artificial selection reveals genetic variation. A locally interbreeding group within a geographic population is called a Mendelian population. Generally, for evolutionary genetic studies, Mendelian populations are considered. To measure genetic variation in a Mendelian population precisely, we would need to count every allele at every locus in every individual in it. By doing so, we could determine the relative proportions, or frequencies, of all alleles in the population. Fortunately, we do not need to make such complete measurements, because we can reliably estimate allele frequencies for a given locus by counting alleles in a sample of individuals from the population. The sum of all allele frequencies at a locus is equal to 1, so measures of allele frequency range from 0 to 1. An allele's frequency is calculated using the following formula. P equals number of copies of the allele in the population by sum of alleles in the population. If only two alleles, for example, A and A, for a given locus are found on autosomes among the members of a diploid population, they may combine to form three different genotypes, AA, AA, and AA. Using the formula above, we can calculate the relative frequencies of alleles A and A in a population of n individuals as follows. Let NAA be the number of individuals that are homozygous for the A allele, AA. Let Na be the number that are heterozygous, AA. Let Na be the number that are homozygous for the A allele, AA. Note that NAA plus Na equals N, the total number of individuals in the population, and that the total number of copies of both autosomal alleles present in the population is 2N because each individual is diploid. Each AA individual has 2 copies of the A allele, and each AA individual has 1 copy of the A allele. Therefore, the total number of A alleles in the population is 2NAA plus Na. Similarly, the total number of A alleles in the population is 2 Na plus Na. If P represents the frequency of A, and Q represents the frequency of A, then. To show how this formula works, Figure 66 calculates allele frequencies in two populations, each containing 200 diploid individuals. Population 1 has mostly homozygotes, 90 AA, 40 AA, and 70 AA. Population 2 has mostly heterozygotes, 45 AA, 130 AA, and 25 AA. Figure 65, calculation of allele frequency in two different populations. The calculations in Figure 65 demonstrate two important points. 1. For each population, P plus Q equals 1. If there is only one allele in a population, its frequency is 1. If an allele is missing from a population, its frequency is 0, and the locus in that population is represented by one or more other alleles. Since P plus Q equals 1, then Q equals 1, P. So when there are only two alleles at a given locus in a population, we can calculate the frequency of one allele and then easily obtain the second alleles. Frequency by subtraction. 2. Both population 1, consisting mostly of homozygotes, and population 2, consisting mostly of heterozygotes, have the same allele frequencies for A and A. Therefore, they have the same gene pool for this locus. However, because the alleles in the gene pool are distributed differently, the genotype frequencies of the two populations differ. Genotype frequencies are calculated as the number of individuals that have the genotype divided by the total number of individuals in the population. In population 1 in Figure 65, the genotype frequencies are 0.45 AA, 0.20 alpha alpha, and 0.35 AA. The frequencies of different alleles at each locus and the frequencies of different genotypes in a Mendelian population describe its genetic structure. Allele frequencies measure the amount of genetic variation in a population. Genotype frequencies show how a population's genetic variation is distributed among its members. These methods for allele frequency and genotype frequency estimation can be used for both non-evolving, Hardy-Weinberg equilibrium, and evolving populations. With these measurements, it becomes possible to consider how the genetic structure of a population changes or does not change over generations. 6.2 The Hardy-Weinberg equilibrium. The Hardy-Weinberg equilibrium is a principle stating that the genetic variation in a population will remain constant from one generation to the next in the absence of disturbing factors and population do not evolve. When mating is random in a large population with no disruptive circumstances, the law predicts that both genotype and allele frequencies will remain constant because they are in equilibrium. Thus, sexual reproduction alone cannot bring evolution. 1908 A British mathematician Godfrey Hardy and a German physician Wilhelm Weinberg tried to find out the standard conditions which are needed to achieve the equilibrium. Hardy wrote his equations in response to a question posed to him by the geneticist Reginald C. Punnett at the Cambridge University Faculty Club. Punnett wondered at the fact that even though the allele for short, stubby fingers, a condition called brachydactyly, was dominant and the allele for normal-length fingers was recessive, most people in Britain have normal-length fingers. Hardy's equations explain why dominant alleles do not necessarily replace recessive alleles in populations, as well as other features of the genetic structure of populations. The Hardy-Weinberg equilibrium applies to sexually reproducing organisms. The particular example we will illustrate here assumes that the organism in question is diploid, its generations do not overlap, the gene under consideration has two alleles, and allele frequencies are identical in males and females. The Hardy-Weinberg equilibrium also applies if the gene has more than two alleles and generations overlap. But in those cases the mathematics is more complicated. Several conditions must be met for a population to be at Hardy-Weinberg equilibrium. Mating is random, no inbreeding, no assortive mating, no mate choice. Population size is very large. There is no migration between populations. There is no mutation. Natural or artificial selection does not affect the alleles under consideration. there is no genetic drift. If these conditions hold, two major consequences follow. 1. The frequencies of alleles at locus will remain a constant from generation to generation. 2. After one generation of random mating, the genotype frequencies will remain in the following proportions. Genotype AA. Frequency P superscript 2 PQ Q superscript 2. Stated another way, the genotype frequency equation for Hardy-Weinberg equilibrium is P superscript 2 plus 2 PQ plus Q superscript 2 equals 1. Figure 66. Calculation of genotype frequency from allele frequency. To see why, consider population 1 in Figure 66, in which the frequency of A alleles, P, is 0.6. Because we assume that individuals select mates at random, without regard to their genotype, gametes carrying A or a combine at random, that is, as predicted by the frequencies P and Q. The probability that a particular sperm or egg in this example will bear an A allele rather than an A allele is 0.6. In other words, 60 out of 100. Randomly sampled sperm or eggs will bear an A allele. Because Q equals 1p, the probability that a sperm or egg will bear an A allele is 1-0.6-0.4. To obtain the probability of two A-bearing gametes coming together at fertilization, we multiply the two independent probabilities of their occurring separately. Pxp equals P superscript 2 equals 0.6, superscript 2 equals 0.36. Therefore, 0.36, or 36%, of the offspring in the next generation will have the AA genotype. Similarly, the probability of bringing together two A-bearing gametes is Q times Q equals Q superscript 2 sad face 0.4, superscript 2 equals 0.16. Thus, 16% of the next generation will have the AA genotype, Figure 66. Figure 66 also shows that there are two ways of producing a heterozygote. An A sperm may combine with an egg, the probability of which is P times Q, or an A sperm may combine with an A egg, the probability of which is Q times P. Consequently, the overall probability of obtaining A heterozygote is 2pq, which will be 2 times 0.6 times 0.4 is equal to 0.48 or 48%. It is now easy to show that the allele frequencies P and Q remain constant for each generation. If the frequency of A alleles in a randomly mating population is P superscript 2 plus PQ, this frequency becomes P superscript 2 plus P, 1p, equals P superscript 2 plus P superscript 2 equals P. The original allele frequencies are unchanged, and the population is at Hardy-Weinberg equilibrium. If allele frequencies are altered due to emigration, the genotype frequencies would automatically settle into a predictable new set in the next generation. For instance, if only AA and AA individuals left the population, P and Q would change, but there would still be AA individuals in the population. 8.2.1 Importance of Hardy-Weinberg equilibrium. Hardy-Weinberg equilibrium suggests that unless some evolutionary agent acts on populations, allele frequencies remain constant from generation to generation. To make sure whether the evolutionary agents are in play, 1 must estimate the actual allele or genotype frequencies present in a population and then compare them with the frequencies that would be expected at Hardy-Weinberg equilibrium under standard conditions. If the obtained values vary from the values obtained from the Hardy-Weinberg expectations, then it suggests which of the standard assumptions are violated. Therefore, the agents of evolutionary change can be identified and one can precisely pay attention towards the same factor for detailed study. 8.2.2 Implications of the Hardy-Weinberg law. Several important implications of the Hardy-Weinberg law for the genetic studies of the population have been noted. 1. That if Hardy-Weinberg assumptions are met, then the population will not evolve, because for evolution to occur, change in the allelic frequencies of a population must take place. Therefore, the Hardy-Weinberg law tells us it's not only reproduction alone but other factors such as natural Selection, mutation, migration, or chances in small populations are also involved in evolution. 2. When a population is in Hardy-Weinberg equilibrium, the genotypic frequencies are determined by the allelic frequencies. For a locus with 2 alleles, the frequency of the heterozygote is greatest when allelic frequencies are between 0.33 and 0.66 and is at a maximum when allelic frequencies are each 0.5. The heterozygote frequency also never exceeds 0.5. Figure 67. Furthermore, heterozygotes for the allele whose frequency is already low will be rarely found in the population, and most of the copies of a rare allele will be present in heterozygotes. As you can see from figure 68, when the frequency of allele A is 0.2, the frequency of the AA homozygote is only 0.04, Q superscript 2, but the frequency of AA heterozygotes is 0.32, 2pq, 80% of the A alleles are in heterozygotes. 3. A single generation of random mating produces the equilibrium frequencies of P superscript 2, 2pq, and q2. If the observed results match to that of Hardy-Weinberg proportions, this simply means only that these forces have not acted since the last time random mating took place, and it does not prove that the population is free from natural selection, mutation, or migration. 2.3. Extension of the Hardy-Weinberg equilibrium. Multiple alleles and X-linked alleles can also be studied with reference to the Hardy-Weinberg law. The genotypic frequencies expected under Hardy-Weinberg equilibrium will differ according to the situation. 8.3.1 Hardy-Weinberg expectations for loci with multiple alleles. Generally, the genotypic frequencies expected at equilibrium are the square of the allelic frequencies. For an autosomal locus with 2 alleles, these frequencies are P plus Q, superscript 2 equals plus 2pq plus Q superscript 2. We can also use the square of the allelic frequencies to calculate the equilibrium frequencies for a locus with multiple alleles. An autosomal locus with 3 alleles, A superscript 1, A superscript 2 and A superscript 3, has 6 genotype. A superscript 1, A superscript 1, A superscript 1, A superscript 2, A superscript 2, A superscript 2, A superscript 1, A superscript 3, A superscript 2, A superscript 3, and A superscript 3, A superscript 3. According to the Hardy-Weinberg law, the frequencies of the genotypes at equilibrium depend on the frequencies of the alleles. If the frequencies of alleles A superscript 1, A superscript 2, and A superscript 3 are P, Q, and R, respectively, then the equilibrium genotypic frequencies will be the square of the allelic frequencies. P plus Q plus R, superscript 2 equals P superscript 2 plus 2pq plus Q superscript 2 plus 2pr plus 2qr plus R superscript 2. Where P superscript 2 equals F, A superscript 1, A superscript 1, 2pq equals F, A superscript 1, A superscript 2, Q superscript 2 equals F, A superscript 2, A superscript 2, 2pr equals F, A superscript 1, A superscript 3, 2qr equals F, A superscript 2, A superscript 3, and R superscript 2 equals F, A superscript 3, A superscript 3. The square of the allelic frequencies can also be used to calculate the expected genotypic frequencies for loci with 4 or more alleles. 8.3.2 Hardy-Weinberg expectations for X-linked loci. For an X-linked locus with 2 alleles, X and X, there are 5 possible genotypes. XAXA, XAX, XX, XAY, and XY, females possess 2 X-linked alleles, and the expected proportions of the female genotypes can be calculated by using the square of the allelic frequencies. If the frequencies of XA and X are P and Q, respectively, then the equilibrium frequencies of the female genotypes are P plus Q, superscript 2 equals P superscript 2, frequency of XAXA equals 2pq, frequency of XAXA equals Q superscript 2, frequency of XX. Males have only a single X-linked allele, and so the frequencies of the male genotypes are P, frequency of XAY, and Q, frequency of XY. Notice that these expected frequencies are the proportions of the genotypes among males and females rather than the proportions among the entire population. Thus, p superscript 2 is the expected proportion of females with the genotype XAXA. If females make up 50% of the population, then the expected proportion of this genotype in the entire population is 0.5x p superscript 2. Genotype frequency p superscript 2 plus 2pq plus q superscript 2 plus p plus q equals 1. The frequency of an X-linked recessive trait among males is q, whereas the frequency among females is q superscript 2. When an X-linked allele is uncommon, the trait will therefore be much more frequent in males than in females. Consider hemophilia A clotting disorder caused by an X-linked recessive allele with a frequency, q, of approximately 1 in 10,000, or 0.0001. At Hardy-Weinberg equilibrium, this frequency will also be the frequency of the disease among males. The frequency of the disease among females, however, will be A(2) sad face 0.0001 equals 0.00000001, which is only 1 in 10 million. Hemophilia is 1,000 times as frequent in males as in females. 8.3.3 Linkage disequilibrium. Consider two different loci on the chromosome, each of which have two alleles. If regular events of genetic recombination occur over a period of time, then the frequencies of the allelic combinations at two syntenic loci will eventually reach to equilibrium. If equilibrium is not reached, the alleles are said to be in linkage disequilibrium. Linkage disequilibrium is the non-random association of alleles at different loci in a given population. Loci are said to be in linkage disequilibrium when the frequency of association of their different alleles is higher or lower than what would be expected if the loci were independent and associated randomly. The effect is due to tendency of two or more linked alleles to be inherited together more often than expected. Such groups of genes have also been referred to as supergenes. 16.4 Practical application of Hardy-Weinberg law. 8.4.1 Complete dominance. When Hardy-Weinberg equilibrium exists, allele frequencies can even be found out in presence of complete dominance where two genotypes cannot be distinguished. If two genotypes AA and AD have the same phenotype due to complete dominance of A over the allele, frequencies can be determined from the frequencies of individuals showing the recessive phenotype AA. The frequency of AA individuals must be equal to the square of the frequency of the recessive allele Q. Let us suppose Q equals 0.5, then Q superscript 2 sad face 0.5, superscript 2 equals 0.25. In other words, when AA phenotype is 0.25 in the population, then it follows that the frequency of the recessive allele A is 0.25 is equal to 0.5. The frequency of the dominant allele A would be 1, Q or 1 minus 0.25 is equal to 0.75. 8.4.2 Frequencies of harmful recessive alleles. The Hardy-Weinberg law can also be used to calculate the frequency of heterozygous carriers of harmful recessive genes. If there are two alleles A and A at an autosomal locus with frequencies P and Q in the population and P plus Q equals 1, then the frequency of AA, and AA genotypes would be P superscript 2 plus 2PQ plus Q superscript 2. If the AA genotype expresses a harmful phenotype such as cystic fibrosis, then the proportion of affected individuals in the population would be Q superscript 2, and the frequency of the heterozygous carriers of the recessive allele would be 2PQ. To illustrate with figures, suppose 1 out of 1,000 children is affected with cystic fibrosis, then the frequency Q superscript 2 equals 0.001, so that Q equals 0.001, which is about 0.032, then 2PQ equals 2 times 0.032 times 0.968 is equal to 0.062. This means that about 62 individuals out of 1,000 or 1 out of 16 is a carrier of the allele for cystic fibrosis. As already mentioned, the number of individuals, AA, who are actually affected is 1 out of 1,000. This implies that the frequency of heterozygous carriers is much higher than that of affected homozygotes. 8.4.3 Multiple alleles. The Hardy-Weinberg law permits calculation of genotypic frequencies at loci with more than 2 alleles, such as the ABO blood groups. There are 3 alleles IA, IB and IO with frequencies P, Q and R. Here P plus Q plus R equals 1. The genotypes of a population with random mating would be P plus Q plus R, superscript 2. Suppose frequency of blood group O in population is 0.16 and rest of 2 alleles, IA and LB, are equally frequent. We can estimate allele frequency of all 3 allele. The person with blood group O represents R superscript 2. So R will be square root of 0.16, i.e. 0.4. Rest of 2 allele R, 1-0.4-0.6, are equally frequent. It means they must be 0.3 each. 8.4.4 Sex-linked loci. It is possible to apply Hardy-Weinberg law for calculating gene frequencies in case of sex-linked loci in males and females. Red-green color blindness is a sex-linked recessive trait. Let denote the recessive allele which produces affected individuals, and R. The normal allele. The frequency of R is P and of R is Q, where P plus Q equals 1. The frequencies of females having RR, RR genotypes would be P2, 2PQ, Q2 respectively. Males are different as they are hemizygous, have only one X chromosome derived from the mother with a single allele, either R or R. The frequency of affected R males would be the same as the frequency of the R allele among the eggs, that is Q. The frequency of normal R males would be P. Suppose the frequency of R alleles is 0.08, then the incidence of affected males would be 0.08 or about 8%. The frequency of affected RR females would be 0.08, 2 equals 0.0064 or 0.64%. Thus the Hardy-Weinberg law explains that males would be affected 100 times more frequently than females. This is actually what is observed. 8.5 Effect of non-random matting on allele frequency. The Hardy-Weinberg law indicates that allelic frequencies do not change as a result of reproduction. Thus, other processes must cause alleles to increase or decrease in frequency. Processes that bring about change in allelic frequency include non-random matting, mutation, migration, genetic drift, random effects due to small population size, and natural selection. An assumption of the Hardy-Weinberg law is that mating is random with respect to genotype. Non-random mating affects the way in which alleles combine to form genotypes and alters the genotypic frequencies of a population. 2 types of non-random mating are 8.5.1. Positive assortative mating refers to a tendency for like individuals to mate. For example, humans exhibit positive assortative mating. For height, tall people mate preferentially with other tall people, short people mate preferentially with other short people. 8.5.2 Negative assortative mating refers to a tendency for unlike individuals to mate. If people engaged in negative assortative mating for height, tall and short people would preferentially mate. 8.5.3 Inbreeding. One form of non-random mating is inbreeding, which is preferential mating between related individuals. Inbreeding is actually positive assortative mating for relatedness, but it differs from other types of assortative mating because it affects all genes, not just those that determine the trait for which the mating preference occurs. Inbreeding causes a departure from the Hardy-Weinberg equilibrium. Frequencies of P superscript 2, 2PQ, and Q superscript 2. More specifically, it leads to an increase in the proportion of homozygotes and a decrease in the proportion of heterozygotes in a population. Outcrossing is the avoidance of mating between related individuals. Inbreeding is usually measured by the inbreeding coefficient, designated F, which is a measure of the probability that 2 alleles are identical by descent. In a diploid organism, homozygous individual has 2 copies of the same allele. These 2 copies may be the same in state. which means that the two alleles are alike in structure and function but do not have a common origin. Alternatively, the two alleles in a homozygous individual may be the same because they are identical by descent. The copies are descended from a single allele that was present in an ancestor. Figure 68. Figure 68, homozygote among the individuals can be due to state or by descent. If we go back far enough in time, many alleles are likely to be identical by descent, but, for calculating the effects of inbreeding, we consider identity by descent by going back only a few generations. Inbreeding coefficients can range from 0 to 1. A value of O indicates that mating in a large population is random. A value of 1 indicates that all alleles are identical by descent. Inbreeding coefficients can be calculated from analyses of pedigrees, or they can be determined from the reduction in the heterozygosity of a population. In weaker forms of inbreeding, such as matings between cousins, the same thing happens, only more slowly. A convenient measure of the effect of inbreeding is based on the reduction in heterozygosity that occurs. Although we will not go into the details of how F is calculated, it's important to understand how inbreeding affects genotypic frequencies. When inbreeding occurs, the frequency of the genotypes will be FAA equals P superscript 2 plus FPQ, FAA equals 2PQ plus 2PQ, and FAA equals Q superscript 2 plus FPQ. With inbreeding, the proportion of heterozygotes decreases by 2 FPQ, and half of this value, FPQ, is added to the proportion of each homozygote. Consider a population that reproduces by self-fertilization, so F equals 1. We will assume that this population begins with genotypic frequencies in Hardy-Weinberg proportions, P superscript 2, 2PQ, and Q superscript 2. With selfing, each homozygote produces progeny only of the same homozygous genotype. AA times AA produces all AA, and AAXAA produces all AA, whereas only half the progeny of a heterozygote will be like the parent. AAXAA produces AA, AA, and AA. Selfing therefore reduces the proportion of heterozygotes in the population by half with each generation, until all genotypes in the population are homozygous. Table 11 and Figure 69. For most outcrossing species, close inbreeding is harmful because it increases the proportion of homozygotes and thereby boosts the probability that deleterious and lethal recessive alleles will combine to produce homozygotes with a harmful trait. Table 11. Generational increases in frequency of homozygous in a self-fertilizing population starting with P equals Q equals 0.5. Assume that a recessive allele, A, that causes a genetic V disease has a frequency, Q, of 0.01. If the population mates randomly, F equals 0, the frequency of individuals affected with the disease, AA, will be Q superscript 2 equals 0.01 superscript 2 equals 0.0001, so only 1 in 10,000 individuals will have the disease. However, if F equals 0.25, the equivalent of brother-sister mating, then the expected frequency of the homozygote genotype is Q superscript 2 plus 2PQF equals 0.01, 2 plus 2, 0.99, 0.01, 0.25, equals 0.0026, thus, the genetic disease is 26 times as frequent at this level of inbreeding. This increased appearance of lethal and deleterious traits with inbreeding is termed inbreeding depression. The more intense the inbreeding, the more severe the inbreeding depression. The harmful effects of inbreeding have been recognized by humans for thousands of years and are the basis of cultural taboos against mating between close relatives. Inbreeding also has deleterious effects on crops, Figure 70, and domestic animals. Figure 70. Deleterious effect on crops increases with inbreeding. Inbreeding depression is most often studied in humans, as well as in plants and animals reared in captivity, but the negative effects of inbreeding may be more severe in natural populations. In spite of the fact that inbreeding is generally harmful for outcrossing species, a Number of plants and animals regularly inbreed and are successful. Inbreeding is commonly used to produce domesticated plants and animals having desirable traits. As stated earlier, inbreeding increases homozygosity, and eventually all individuals in the population become homozygous for the same allele. If a species undergoes inbreeding for a number of generations, many deleterious recessive alleles are weeded out by natural or artificial selection so that the population becomes homozygous for beneficial alleles. In this way, the harmful effects of inbreeding may eventually be eliminated, leaving a population that is homozygous for beneficial traits. 8.6 Effect of mutation on allele frequency. Mutation can influence the rate at which one genetic variant increases at the expense of another. Consider a single locus in a population of 25 diploid individuals. Each individual possesses 2 alleles at the locus under consideration, so the gene pool of the population consists of 50 allelic copies. Let us assume that there are 2 different alleles, designated G superscript 1 and G superscript 2 with frequencies P and Q, respectively. If there are 45 copies of G superscript 1, 5 copies of G superscript 2 in the population, P equals 0.90 and Q equals 0.10. Now suppose that a mutation changes a G superscript 1 allele into a G superscript 2 allele. After this mutation, there are 44 copies of G superscript 1 and 6 copies of G superscript 2, and the frequency of G superscript 2 has increased from 0.10 to 0.12. Mutation has changed the allelic frequency. If copies of G superscript 1 continue to mutate to G superscript 2, the frequency of G(2) will increase and the frequency of G superscript 1 will decrease. Figure 71. Figure 71. Change in allelic frequency due to recurrent mutations. The amount that G superscript 2 will change, delta Q, as a result of mutation depends on 1. The rate of G superscript 1, 2. G superscript 2 mutation, U, and 2. P, the frequency of G1 in the population. When P is large, there are many copies of G superscript 1 available to mutate to G superscript 2 and the amount of change will be relatively large. As more mutations occur and P decreases, there will be fewer copies of G superscript 1 available to mutate to G superscript 2. The change in G superscript 2 as a result of mutation equals the mutation rate times the allelic frequency. Delta Q equals mu P. As the frequency of P decreases as a result of mutation, the change in frequency due to mutation will be less, and so far we have considered only the effects of G superscript 1, G superscript 2 forward mutations. Reverse G superscript 2, G superscript 1 mutations also occur at rate, which will probably be different from the forward mutation rate, mu. Whenever a reverse mutation occurs, the frequency of G superscript 2 decreases and the frequency of G superscript 1 increases. Figure 72. The rate of change due to reverse mutations equals the reverse mutation rate times the allelic frequency of G superscript 2, delta Q equals VQ. The overall change in allelic frequency is a balance between the opposing forces of forward mutation and reverse mutation. Delta Q equals mu PVQ. 8.6.1 Mutational equilibrium of allelic frequency. Consider an allele that begins with a high frequency of G superscript 1 and a low frequency of G superscript 2. In this population, many copies of G superscript 1 are initially available to mutate to G superscript 2, and the increase in G superscript 2 due to forward mutation will be relatively large. However, as the frequency of G superscript 2 increases as a result of forward mutations, fewer copies of G superscript 1 are available to mutate, so the number of forward mutations decreases. On the other hand, few copies of G superscript 2 are initially available to undergo a reverse mutation TOG superscript 1, but as the frequency of G superscript 2 increases, the number of copies of G superscript 2 available to undergo reverse mutation to G superscript 1 increases G superscript 1, so the number of genes undergoing reverse mutation will increase. Eventually, the number of genes undergoing forward mutation will be counterbalanced by the number of genes undergoing reverse mutation. At this point, the increase in Q due to forward mutation will be equal to the decrease in Q due to reverse mutation, and there will be no net change in allelic frequency, Q equals 0, in spite of the fact that forward and reserve mutations continue to occur. The point at which there is no change in the allelic frequency of population is referred to as equilibrium, figure 72. 8.6.2 Factors determining allelic frequencies at equilibrium. We can determine the allelic frequencies at equilibrium by manipulating equation 1. Recall that P1 equals Q. Substituting 1 Q for P in equation 1, we get where and Q equals the frequency of G(2) at equilibrium. This final equation tells us that the allelic frequency at equilibrium is determined solely by the forward and reverse mutation rates. Thus, when the only evolutionary force acting on a population is mutation, allelic frequencies change with the passage of time because some alleles mutate into others. Eventually, these allelic frequencies reach equilibrium and are determined only by the forward and reverse mutation rates. When the allelic frequencies reach equilibrium, the Hardy-Weinberg law tells us that genotypic frequencies also will remain the same. The mutation rates for most genes are low, so change in allelic frequency due to mutation in one generation is very small, and long periods of time are required for a population to reach mutational equilibrium. For example, if the forward and reverse mutation rates for alleles at a locus are 1 times 10 and 0.3 times 10 per generation, respectively, rates that have actually been measured at several loci in mice, and the allelic frequencies are P equals 0.9 and Q equals 0.1, then the net change in allelic frequency per generation due to mutation is delta Q equals mu nu Q. Sad face 1 times 10 dash 0.9 dash 0.3 times 10, 0.1 equals 8.7 x 10 equals 0.000087. Therefore, change due to mutation in a single generation is extremely small and, as the frequency of P drops as a result of mutation, the amount of change will become even smaller, figure 72. In an experiment it was observed that the rate of mutation in E. coli from lac to lac plus are determined using medium containing lactose, as the only sole source of energy. The rate of lac to lac mutation in the presence of lactose was similar to overall rate of mutation at any other locus. This simply suggests that mutations are spontaneous. Figure 72. Change due to recurrent mutations slows as frequency of P decreases with number of generations. Consider an autosomal locus with two alleles A1 and A2 at frequencies of 0.9 and 0.1 respectively as mentioned above. Each generation, A1 mutates to A2 at a rate of mu equals 1 times 10 while A2 mutates to A1 at a rate of u equals 0.3 times 10. Assume that the population is infinitely large and no other evolutionary force is acting. The equilibrium frequency of allele A2 is. The effect of typical mutation rates on Hardy-Weinberg equilibrium is negligible, and many generations are required for a population to reach mutational equilibrium. Nevertheless, if mutation is the only force acting on a population for long periods of time, mutation rates will determine allelic frequencies. 8.7 Effect of migration on allele frequency. Another process that may bring about change in the allelic frequencies is the influx of genes from other populations, commonly called migration or gene flow. One of the assumptions of the Hardy-Weinberg law is that migration does not take place, but many natural populations do experience migration from other populations. The overall effect of migration is twofold. 1. It prevents genetic divergence between populations and 2. It increases genetic variation within populations. The effect of migration on allelic frequencies. Let us consider the effects of migration by looking at a simple, unidirectional model of Migration between two populations that differ in the frequency of an allele A, say the frequency of this allele in population I is Chi and in population 2 is Qi, figure 73a and b. In each generation, a representative sample of the individuals in population I migrates to population 2, figure 73c, and reproduces, adding its genes to population 2's gene pool. Migration is only from population I to population 2, is unidirectional, and all the conditions of the Hardy-Weinberg law apply, except the absence of migration. Conclusion: the frequency of allele in population 2 after migration is figure 73. The amount of change in allelic frequency due to migration between populations depends on the difference in allelic frequency and exteny of migration. After migration, population 2 consists of two types of individuals, figure 73d. Some are migrants, they make up proportion M of population 2, and they carry genes from population I, so the frequency of allele in the migrants is Chi. The other individuals in population 2 are the original residents. If the migrants make up proportion M of population 2, then the residents make up 1M because the residents originated in population 2, the frequency of allele in this group is. After migration, the frequency of allele in the merged population. The overall effect of migration. It causes the gene pools of populations to become more similar. Later, we will see how genetic drift and natural selection lead to genetic differences between populations. Migration counteracts this tendency and tends to keep populations homogeneous in their allelic frequencies. 2. Migration adds genetic variation to populations. Different alleles may arise in different populations owing to rare mutational events, and these alleles can be spread to new populations by migration, increasing the genetic variation within the recipient population. 6.8 Genetic drift. The Hardy-Weinberg law assumes random mating in an infinitely large population. Only when population size is infinite will the gametes carry genes that perfectly represent the parental gene pool. But no real population is infinitely large, and when population size is limited, the gametes that unite to form individuals of the next generation carry a sample of alleles present in the parental gene pool. Just by chance, the composition of this sample may deviate from that of the parental gene pool, and this deviation may cause allelic frequencies to change. The smaller the gametic sample, the greater the chance that its composition will deviate from that of the entire gene pool. The role of chance in altering allelic frequencies is analogous to flipping a coin. Each time we flip a coin, we have a 50% chance of getting a head and a 50% chance of getting a tail. If we flip a coin 1,000 times, the observed ratio of heads to tails will be very close to the expected 50-50 ratio. If, however, we flip a coin only 10 times, there is a good chance that we will obtain not exactly 5 heads and 5 tails, but rather maybe 7 heads and 3 tails or 8 tails and 2 heads. This kind of deviation from an expected ratio due to limited sample size is referred to as sampling error. Sampling error occurs when gametes unite to produce progeny. Many organisms produce a large number of gametes, but, when population size is small, a limited number of gametes unite to produce the individuals of the next generation. Chance influences which alleles are present in this limited sample and, in this way, sampling error may lead to changes in allelic frequency, which is called genetic drift. Because the deviations from the expected ratios are random, the direction of change is unpredictable. We can nevertheless predict the magnitude of the changes. 8.8.1 The magnitude of genetic drift. The amount of sampling error resulting from genetic drift can be estimated from the variance in allelic frequency. Variance is a statistical measure that describes the degree of variability in a trait. Suppose that we observe a large number of separate populations, each with n individuals and allelic frequencies of P and Q. After one generation of random mating, genetic drift expressed in terms of the variance in Allelic frequency among the populations, SP superscript 2, will be SP superscript 2 equals PQ/2N. The amount of change resulting from genetic drift, the variance in allelic frequency, is determined by two parameters, the allelic frequencies P and Q, and the population size, N. Genetic drift will be maximal when P and Q are equal, each 0.5, and when the population size is small. For ecological and demographic studies, population size is usually defined as the number of individuals in a group. The evolution of a gene pool depends, however, only on those individuals who contribute genes to the next generation. Population geneticists usually define population size as the equivalent number of breeding adults, the effective population size, Na. Several factors determine the equivalent number of breeding adults. One factor is the sex ratio. When the numbers of males and females in the population are equal, the effective population size is simply the sum of reproducing males and females. When they are unequal, then the effective population size is. The sex ratio is the ratio of the number of males to the number of females. Table 12 gives the effective population size for a theoretical population of 100 individuals with different proportions of males and females. Notice that, when the number of males and females is unequal, the effective population size is smaller than it is when the number of males and females is the same. For example, when a population consists of 90 males and 10 females, the effective population size is only 36, and genetic drift will occur as though the actual population consisted of only 36 individuals, equally divided between males and females. A population with 90 males and 10 females has the same effective population size as a population with 10 males and 90 females. It makes no difference which sex is in excess. The reason that the sex ratio influences genetic drift is that half the genes in the gene pool come from males and half come from females. When one sex is present in low numbers, genetic drift increases because half of the genes are coming from a small number of individuals. In a population consisting of 10 males and 90 females, the overall population size is relatively large, 100, but only 10 males contribute half the genes to the next generation. Sampling error therefore affects the range of genes present in the male gametes, and chance will have a major effect on the allelic frequencies of the next generation. Other factors that influence effective population size include variation between individuals in reproductive success, fluctuations in population size, the age structure of the population, and whether mating is random. 8.8.2 Causes of genetic drift. Certain circumstances can result in genetic drift having a significant impact on a population. Two examples are the founder effect and the bottleneck effect. 1. The founder effect. When a few individuals become isolated from a larger population, this smaller group may establish a new population whose gene pool differs from the source population. This is called the founder effect. The founder effect might occur, for example, when a few members of a population are blown by a storm to a new island. Genetic drift, in which chance events alter allele frequencies, will occur in such a case if the storm indiscriminately transports some individuals, and their alleles, but not others, from the source population. The founder effect probably accounts for the relatively high frequency of certain inherited disorders among isolated human populations. 2. The bottleneck effect, a sudden change in the environment, such as a fire or flood, may drastically reduce the size of a population. A severe drop in population size can cause the bottleneck effect, so named because the population has passed through a bottleneck that reduces its size. Figure 75. Figure 75. The genetic drift due to bottleneck effect. By chance alone, certain alleles may be overrepresented among the survivors, others may be underrepresented, and some may be absent altogether. Ongoing genetic drift is likely to have substantial effects on the gene pool until the population becomes large enough that chance events have less impact. But even if a population that has passed through a bottleneck ultimately recovers in size, it may have low levels of genetic variation for a long period of time, a legacy of the genetic drift that occurred when the population was small. One reason it is important to understand the bottleneck effect is that human actions like overexploitation and hunting sometimes create severe bottlenecks for other species. 8.8.3 The effects of genetic drift. Genetic drift has several important effects on the genetic composition of a population. Genetic drift leads to abrupt change in allelic frequencies within a population. Because drift is random, allelic frequency is just as likely to increase as it is to decrease and will wander with the passage of time, hence the name genetic drift. Figure 76 illustrates a computer simulation of genetic drift in 5 populations over 30 generations, starting with G equals 0.5 and maintaining a constant population size of 10 males and 10 females. These allelic frequencies change randomly from generation to generation. Figure 76. Genetic drift leads to abrupt change in allele frequency in successive generation, which leads to decreased genetic variation through fixation of allele and genetic divergence among populations. 2. Genetic drift reduces genetic variation within populations. Through random change, an allele may eventually reach a frequency of either 1 or 0, at which point all individuals in the population are homozygous for one allele. When an allele has reached a frequency of 1, we say that it has reached fixation. Other alleles are lost, reach a frequency of 0, and can be restored only by migration from another population or by mutation. Fixation, then, leads to a loss of genetic variation within a population. This loss can be seen in northern elephant seals. Today, these seals have low levels of genetic variation. A study of 24 protein encoding genes found no individual or population differences in these genes. Given enough time, all small populations will become fixed for one allele. Which allele becomes fixed is random and is determined by the initial frequency of the allele. If the population begins with 2 alleles, each with a frequency of 0.5, both alleles have an equal probability of fixation. However, if one allele is initially common with frequency of 0.95, it is more likely, 95%, to become fixed. 3. Genetic drift leads to divergence in different populations with time. In Figure 76, all 5 populations begin with the same allelic frequency. Q equals 0.5, but, because drift occurs randomly, the frequencies in different populations do not change in the same way, and so populations gradually acquire genetic differences. Notice that, although the variance in allelic frequency among the populations increases, the average allelic frequency remains basically the same. Eventually, all the populations reach fixation. Some will become fixed for one allele and others will become fixed for the alternative allele. Figure 77. Fixation of alleles. 8.8.4 Changes in the probability that an allele will have various possible frequencies. Proportion of populations with different allele frequencies after t equals 2n generations, for example, after 50 generations, in several population. Each of size n equals 10, proportion of populations with different allele frequency after t equals 2 times 10 times 50 is equal to 1000. Proportion of populations in which allele is fixed or lost increases at a rate of 1/4 n per generation. For example, in several populations, each of size n equals 10, the rate of increase per generation in the proportion of populations in which the allele is lost or fixed would be 1 divided by 40 is equal to 0.025. Each class of allele frequencies between 0 and 1 decrease at a rate of 1/2n per generation, Figure 77. For example, in several populations, each of size n equals 10, the rate of decrease per generation in each allele frequency class between 0 and 1 would be 1 divided by 20 is equal to 0.05. If random genetic drift is the only mechanism acting on these populations, then, after a large number of generations, the expected number of diploid populations fixed for the A allele is number of independent population /2n. For example, 100 independent populations of Drosophila are established with 10 individuals in each population, of which one individual is of AA genotype and the other 9 are of AA genotype. If random genetic drift is the only mechanism acting on these populations, then, after a large number of generations, the expected number of populations fixed for the A allele would be 100/2x10 equals 100 divided by 20 is equal to 5. The three results of genetic drift, allelic frequency change, loss of variation within populations, and genetic divergence between populations, occur simultaneously, and all result from sampling error. The first two results occur within populations, whereas the third occurs between populations. 8.8.5 Summary of effects of genetic drift. The examples we have considered so far highlight following 4 key points. 1. Genetic drift is significant in small populations. Chance events can cause an allele to be disproportionately over or underrepresented in the next generation. Although chance events occur in populations of all sizes, they tend to alter allele frequencies substantially only in small populations. 2. Genetic drift can cause allele frequencies to change at random. Because of genetic drift, an allele may increase in frequency 1 year, then decrease the next. The change from year to year is not predictable. Thus, unlike natural selection, which in a given environment consistently favors some alleles over others, genetic drift causes allele frequencies to change at random over time. 3. Genetic drift can lead to a loss of genetic variation within populations, by causing allele frequencies to fluctuate randomly over time. Genetic drift can eliminate alleles from a population. Because evolution depends on genetic variation, such losses can influence how effectively a population can adapt to a change in the environment. 4. Genetic drift can cause harmful alleles to become fixed. Alleles that are neither harmful nor beneficial can be lost or become fixed entirely by chance through genetic drift. In very small populations, genetic drift can also cause alleles that are slightly harmful to become fixed. When this occurs, the population's survival can be threatened. 6.9 Mutation and drift, the neutral theory. The neutral theory of molecular evolution tries to explain observed patterns in nucleotide frequencies across populations with only two evolutionary forces, mutation and drift. Mutation introduces new variation, and drift causes them to spread. We have already seen that drift can lead to divergence among populations if there is some level of variation initially available. We now assume that all new variation is introduced by mutation and establish 3 important results. 8.9.1 The fixation probability of a neutral mutation is 1/2 n, the probability of an individual being homozygous in a population of finite size n. By random sampling alone, is the probability of getting any gene copy as one of the alleles in a diploid genotype equals 1, and the probability of getting the same gene copy by random sampling equals 1/2n. That is, for any particular individual in a randomly mating population of finite size nf equals 1/2n. The probability of an individual in this population being heterozygous for these gene copies is thus 1f equals 1-1/2n. 8.9.2 The rate of neutral substitution is equal to the neutral mutation rate. Next, we want to calculate the neutral rate of substitution K, defined as the number of all mutations that arise in a population times the probability that any of those mutations is fixed. If the mutation rate per site and generation is mu-omicron, 2 new mu-omicron mutations will arise every generation at the site. We thus have K equals 2 new mu-omicron, 1/2 equals mu-omicron. The rate of substitution is just equal to the rate of new mutations, independent of the population size. This is one of the most famous and most useful results from population genetics. It implies that molecular evolution at a neutral site occurs at an approximately constant rate. per unit time. It is therefore said to show a molecular clock. As a consequence, the number of substitutions between two species can be used to infer the time since these species split from their common ancestor. Importantly, the molecular clock is independent of fluctuations in population size. It assumes, however, that mutation rates are constant over time. 8.9.3 The heterozygosity H assumes a predictable equilibrium value. Neutral theory not only allows us to look at the long-term rate of evolution, but also allows us to predict the amount of genetic variation we expect at any given time. We have already derived that genetic drift reduces the heterozygosity each generation by ADH equals 1/2 NH. Clearly, mutation will increase genetic variation. We can therefore imagine that we reach an equilibrium point where the decrease in H due to drift is balanced by the increase due to mutation drift equilibrium. In order to derive this equilibrium frequency, we first derive the change of H under mutation alone. For an infinite population, the heterozygosity in the new generation before mutation is equal to the heterozygosity in the parent generation, H*prime equals H, Hardy-Weinberg. We now assume that every new mutation results in a new allele that has not been present in the population before, so-called infinite alleles model. This is realistic if we distinguish alleles of a gene on the DNA sequence level. Then every pair of genes with unequal alleles before mutation will also have unequal alleles after mutation. Every pair of genes with equal alleles before mutation will become heterozygote if either of the genes mutates. Summing over these two cases and ignoring terms proportional /mu(2) i.e. that both genes mutate, we obtain. Thus, in a population of effective population size n with rate of neutral mutation mu0, the frequency of heterozygotes per nucleotide per site at equilibrium between mutation and genetic drift, H, is calculated as expected, the equilibrium heterozygosity increases with increasing mutation rate and increasing population size, i.e. reduced drift. 4.10 Natural selection. A final process that brings about changes in allelic frequencies is natural selection, the differential reproduction of genotypes. Natural selection takes place when individuals with adaptive traits produce more offspring. If the adaptive traits have a genetic basis, they are inherited by the offspring and appear with greater frequency in the next generation. A trait that provides a reproductive advantage thereby increases over time, enabling populations to become better suited to their environments to become better adapted. Natural selection is unique among evolutionary forces in that it promotes adaptation. 810.1 Natural selection results in adaptation. The evolutionary agents we have just discussed influence the frequencies of alleles and genotypes in populations. Major perturbations, such as colliding continents, volcanic eruptions, and meteorite impacts, also have periodically altered the survival and reproductive rates of organisms. All of these agents dramatically affect the course of life's evolution on Earth, but none of them result in adaptations. For adaptation to occur, individuals that differ in heritable traits must survive and reproduce with different degrees of success. When some individuals contribute more offspring to the next generation than others, allele frequencies in the population change in a way that adapts individuals to the environments that influence their success. This process is known as natural selection. The reproductive contribution of a phenotype to subsequent generations relative to the contributions of other phenotypes is called its fitness. The word relative is critical. The absolute number of offspring produced by an individual does not influence the genetic structure of a population. Changes in absolute numbers of offspring are responsible for increases and decreases in the size of a population, but only the relative success of different phenotypes within a population leads to changes in allele frequencies, that is, to evolution. To contribute genes to subsequent generations, individuals must survive to reproductive age and produce offspring. The relative contribution of individuals of a particular phenotype is determined by the probability that those individuals survive multiplied by the average number of offspring they produce over their lifetimes. In other words, the fitness of a phenotype is determined by the average rates of survival and reproduction of individuals with that phenotype. 8.10.2 Fitness and Selection Coefficient. The effect of natural selection on the gene pool of a population depends on the fitness values of the genotypes in the population. Fitness is defined as the relative reproductive success of a genotype. Here the term relative is critically important. Fitness is the reproductive success of one genotype compared with the reproductive successes of other genotypes in the population. Fitness, W, ranges from 0 to 1. Suppose the number of viable offspring produced by 3 genotypes is. To calculate fitness for each genotype, we take the average number of offspring produced by a genotype and divide it by the mean number of offspring produced by the most prolific genotype. The fitness of the genotype A superscript 1 is designated W11, that of A superscript 1 is W12, and that of A superscript 2 is W22. A related variable is the selection coefficient, S, which is the relative intensity of selection against a genotype. The selection coefficient is equal to 1 W, so the selection coefficients for the preceding 3 genotypes are 810.3. The results of natural selection. The results of selection depend on the relative fitness of the genotypes. If we have 3 genotypes, A superscript 1, A superscript 1, A superscript 1, A superscript 2, and A superscript 2 with fitness W11, W12 and W22, we can identify 6 different types of natural selection, Table 13. 1. Directional selection. Directional selection favors individuals at one extreme of a phenotypic distribution that have greater reproductive success in a particular environment. One way that directional selection may arise is that a new allele may be introduced into a population by mutation, and the new allele may confer a higher fitness in individuals that carry it. Figure 78. Figure 78. Directional selection selects for a darker phenotype that confers higher fitness, for example, in a polluted environment as with the peppered moth Bestun bechellaria. We have already discussed that how darker color conferred greater fitness to peppered moths in polluted environments. If the homozygote carrying the favored allele has the highest fitness value, directional selection may cause this favored allele to eventually become predominant in the population. Another example of directional selection can be taken from one of Galapagos Islands known as Daphne Major. This small island, 0.3 square kilometers, has a resident population of the medium ground finch, Geospiza fortis. The medium ground finch has a relatively small crushing beak, allowing it to feed on small, tender seeds. The researchers quantified beak size among the medium ground finches of Daphne Major by carefully measuring beak depth, a measure of the beak from top to bottom. They compared the beak sizes of parents and offspring by examining broods over many years. The depth of the beak was inherited by offspring from parents, regardless of environmental conditions, indicating that differences in beak sizes are due to genetic differences in the population. This means that beak depth is a heritable trait. In the wet year of 1976, the plants of Daphne Major produced an abundance of small seeds that finches could easily eat. 2 years later, in 1978, a drought occurred and plants produced few of the smaller seeds and only larger drier seeds, which are harder to crush, were readily available. As a result, birds with larger beaks were more likely to survive because they were better at breaking open the large seeds. In the year after the drought, the average beak depth of birds in the population increased almost 10%. 2. Stabilizing selection. Stabilizing selection favors the survival of individuals with intermediate phenotypes. The extreme values of a trait are selected against. An example of stabilizing selection involves clutch size, the number of eggs laid, in animals. Birds that lay too many or too Few eggs per nest have lower fitness values than do those that lay an intermediate number of eggs. Laying too many eggs is disadvantageous because many chicks die due to an inadequate supply of food. In addition, the parent's survival may be reduced because of the strain of trying to feed a large brood. On the other hand, having too few offspring results in the contribution of relatively few individuals to the next generation. Therefore, an intermediate clutch size is favored. Figure 79. Figure 79. In stabilizing selection, the extremes of a phenotypic distribution are selected against while individuals with intermediate traits are favored. For example, clutch size of birds. 3. Balancing selection. Balancing selection is a type of natural selection that maintains genetic diversity in a population. In balancing selection, two or more alleles are kept in balance, and therefore are maintained in a population over the course of many generations. Balancing selection does not favor one particular allele in the population. This type of selection includes heterozygote advantage and frequency-dependent selection. 4. Heterozygote advantage. Here the heterozygote has higher fitness than the fitnesses of the two homozygotes. With heterozygote advantage or overdominance, both alleles are favored in the heterozygote, and neither allele is eliminated from the population. Initially, the allelic frequencies may change because one homozygote has higher fitness than the other. The direction of change will depend on the relative fitness values of the two homozygotes. Note that heterozygote advantage is defined in terms of genotype, not phenotype. Thus, whether heterozygote advantage represents stabilizing or directional selection depends on the relationship between the genotype and the phenotype. For example, if the phenotype of a heterozygote is intermediate to the phenotypes of both homozygotes, heterozygote advantage is a form of stabilizing selection. A classic example of heterozygote advantage involves the sickle cell allele of the human S-set globin gene in African population where malaria is quite common. A homozygous individual with two copies of this allele has sickle cell disease, a hereditary disease that damages blood cells. The sickle cell homozygote has a lower fitness than a homozygote with two copies of the normal and more common globin allele. However, in areas where malaria is endemic, the heterozygote has the highest level of fitness compared with normal homozygotes. Heterozygotes have 10 to 15% better chance of survival if infected by the malarial parasite, Plasmodium falciparum. Therefore, the sickle cell allele is maintained in populations where malaria is prevalent, even though the allele is detrimental in the homozygous state. Frequency-dependent selection. Frequency-dependent selection is another mechanism that causes balancing selection. Frequency-dependent selection describes the condition where the fitness of one phenotype is dependent on its frequency relative to other phenotypes in the population. In negative frequency-dependent selection, rare phenotypes are favored over common phenotypes. For example, many species of invertebrates exist as different, colored forms, identical in all respects except color. Visually searching predators often develop a search image for one color form, usually the commoner. The prey then proliferates in the rarer form until this form itself becomes more common. In positive frequency-dependent selection, common phenotypes have an advantage. For example, where prey are warningly colored to advertise bad taste or toxicity, the prey gain the most benefit where many individuals have warning coloration and the predator is deterred. 4. Disruptive selection. Disruptive selection or underdominance favors the survival of individuals at both extremes of a range, rather than the intermediate. Underdominance leads to an unstable equilibrium. Here allelic frequencies will not change as long as they are at equilibrium, but, if they are disturbed from the equilibrium point by some other evolutionary force, they will move away from equilibrium until one allele eventually becomes fixed. It is similar but not identical to balancing selection where individuals of average trait values are favored against those of extreme trait values. The fitness values of one genotype are higher in one environment, while the fitness values of the other genotype are higher in another. Environment. An example in favor of F disruptive selection can be seen in colonial bentgrass, Agrostis tenuis. In certain locations where this grass is found, isolated places occur where there are high levels of heavy metals such as copper from mining. Such pollution has selected for mutant strains that show tolerance to copper, figure 80. This genetic change enables the plants to grow on copper-contaminated soil but tends to inhibit growth on a normal, uncontaminated soil. This results in metal-resistant plants growing on contaminated sites that are close to normal plants growing on uncontaminated land. Figure 80, disruptive selection. In this example, the normal wild-type colonial bent grass, Agrostis tenuis, is intolerant to metals in the soil. A mutation creates a metal-tolerant variety, which can grow in soils contaminated with metals from mining operations. 5. Experimental observation and inference. Generally, the body weight of adult female of a strain of Drosophila is around 1.8 plus or minus 0.45 mg, mean plus or minus standard deviation. Suppose in a laboratory experiment, each of the three groups, A, B, C, of this strain was subjected to a different type of selection pressure having influence on the female body weight. After many generations of experimental selection pressure, the body weight changed as follows, figure 81. Group A, distribution, normal, 1.8 plus or minus 0.08 milligrams. Group B, distribution, normal, 2.2 plus or minus 0.08 milligrams. Group C, distribution, bimodal at 1.4 and 2.2 mg plus or minus 0.08. From this experiment it can be concluded that Group A is under stabilizing selection as mean is on same position, while Group B has undergone where one of the extreme is being selected. In Group C, both the extreme variants have been selected, which represents disruptive selection. Figure 81, different experimental results after selection. The rate at which an allele changes in frequency owing to selection depends on the intensity of selection and the dominance relations among the genotypes, figure 82. Under directional selection, dominant alleles will increase much more rapidly than recessive alleles, because homozygotes and heterozygotes are favored. With incomplete dominance, the heterozygote has a selective advantage, but not as much as the homozygote, so incompletely dominant alleles increase in frequency at a lower rate than that of dominant alleles. Recessive alleles increase at the lowest rate because only the homozygote is favored by selection. The rate at which selection changes allelic frequencies also depends on the allelic frequency itself. If an allele, A2, is lethal and recessive, W11 equals W12-1, whereas W22-0. The frequency of the A superscript 2 allele will decrease over time, because Thea A superscript 2 homozygote produces no offspring. And the rate of decrease will be proportional to the frequency of the recessive allele. When the frequency of the allele is high, the change in each generation is relatively large, but, as the frequency of the allele drops, a higher proportion of the alleles are in the heterozygous genotypes, where they are immune to the action of natural selection. The heterozygotes have the same phenotype as the favored homozygote. Thus, selection against a rare recessive allele is very inefficient and its removal from the population is slow. Figure 82. The rate of change of allelic frequency data due to selection depends on the dominance relations among the genotype. The relation between the frequency of a recessive allele and its rate of change under natural selection has an important implication. Some people believe that the medical treatment of patients with rare recessive diseases will cause the disease gene to increase, eventually leading to degeneration of the human gene pool. This mistaken belief was the basis of eugenic laws that were passed in the early part of the 20th century, prohibiting the marriage of persons with certain genetic conditions and allowing the involuntary sterilization of others. However, most copies of rare recessive alleles are present in heterozygotes, and selection against the homozygotes will have little effect on the frequency of a recessive allele. Thus, whether the homozygotes reproduce or not has little effect on the frequency of the disorder. 8.10.4 Natural selection cannot lead to perfect organisms. Though natural selection leads to adaptation, nature abounds with examples of organisms that are less than ideally engineered for their lifestyles. There are several reasons why. 1. Selection can act only on existing variations. Natural selection favors only the fittest phenotypes among those currently in the population, which may not be the ideal traits. New advantageous alleles do not arise on demand. 2. Evolution is limited by historical constraints. Each species has a legacy of descent with modification from ancestral forms. Evolution does not scrap the ancestral anatomy and build each new complex structure from scratch. Rather, evolution co-opts existing structures and adapts them to new situations. We could imagine that if a terrestrial animal were to adapt to an environment in which flight would be advantageous, it might be best just to grow an extra pair of limbs that would serve as wings. However, evolution does not work this way. Instead, it operates on the traits an organism already has. Thus, in birds and bats, an existing pair of limbs took on new functions for flight as these organisms evolved from non-flying ancestors. 3. Adaptations are often compromises. Each organism must do many different things. A seal spends part of its time on rocks. It could probably walk better if it had legs instead of flippers, but then it would not swim nearly as well. We humans owe much of our versatility and athleticism to our prehensile hands and flexible limbs, but these also make us prone to sprains, torn ligaments, and dislocations. Structural reinforcement has been compromised for agility. For chance, natural selection and the environment interact. Chance events can affect the subsequent evolutionary history of populations. 4. Instance, when a storm blows insects or birds hundreds of kilometers over an ocean to an island, the wind does not necessarily transport those individuals that are best suited to the new environment. Thus, not all alleles present in the founding population's gene pool are better suited to the new environment than the alleles that are left behind. In addition, the environment at a particular location may change unpredictably from year to year, again limiting the extent to which adaptive evolution results in a close match between the organism and current environmental conditions. With these 4 constraints, evolution does not tend to craft perfect organisms. 9.11 Mutation and natural selection. Recurrent mutation and natural selection act as opposing forces on detriment alleles. Mutation increases their frequency and natural selection decreases their frequency. Eventually, these two forces reach an equilibrium, in which the number of alleles added by mutation is balanced by the number of alleles removed by selection. The change in allelic frequency due to selection against a recessive allele is SPQ superscript 2/1 SQ superscript 2. When Q is very low, Q superscript 2 is near 0, so 1 SQ superscript 2 will be approximately 1 minus 0 is equal to 1. Thus, when Q is very low, the decrease in frequency due to selection is approximately SPQ superscript 2. The increase in frequency of an allele due to forward mutations is P. At equilibrium, the effects of mutation and selection are balanced, so SPQ superscript 2 equals up. This equation can be rearranged. Q superscript 2 equals up equals U. SPS. Taking the square root of each side, we get the frequency of the allele at equilibrium, Q, is therefore equal to the square root of the mutation rate divided by the selection coefficient. With the use of the equation for selection acting on a dominant allele and similar reasoning, the frequency of a dominant allele at equilibrium can be shown to be. Achondroplasia is a common type of human dwarfism that results from a dominant gene. People with this condition are fertile, although they produce only about 74% as many children as are produced by people without achondroplasia. The fitness of people with achondroplasia Therefore averages 0.74, and the selection coefficients is 1, W, or 0.26. If we assume that the mutation rate for achondroplasia is about 3 times 10, a typical mutation rate in humans, then we can predict that the equilibrium frequency for the achondroplasia allele will be equals Q equals 0.00003/0.26 equals 0.00011153. This frequency is close to the actual frequency of the disease. 8.12 The general effects of evolutionary forces. The 4 important processes bring about change in the allelic frequencies of a population are mutation, migration, genetic drift, and natural selection in short term. Their long-term effects on allelic frequencies are summarized in Table 14. In some cases, these changes continue until one allele is eliminated and the other becomes fixed in the population. Genetic drift and directional selection will eventually result in fixation, provided these forces are the only ones acting on a population. With the other evolutionary forces, allelic frequencies change until an equilibrium point is reached, and then there is no additional change in allelic frequency. Mutation, migration, and some forms of natural selection can lead to stable equilibria. Table 14, long-term effects of different evolutionary forces on allelic frequencies within populations. The different evolutionary forces affect both genetic variation within populations and genetic divergence between populations. Evolutionary forces that maintain or increase genetic variation within populations are listed in the upper left quadrant of Figure 83 and that decrease genetic variation within populations are listed in the lower left quadrant of Figure 83. These forces include genetic drift, which decreases variation through the fixation of alleles, and some forms of natural selection such as directional selection. The various evolutionary forces also affect the amount of genetic divergence between populations. Natural selection increases divergence between populations if different alleles are favored in the different populations, but it can also decrease divergence between populations by favoring the same allele in the different populations. Mutation almost always increases divergence between populations because different mutations arise in each population. Genetic drift also increases divergence between populations because changes in allelic frequencies due to drift are random and are likely to change in different directions in separate populations. Migration, on the other hand, decreases divergence between populations because it makes populations similar in their genetic composition. Figure 83, influence of different evolutionary forces on genetic variation within and between populations. Migration and genetic drift act in opposite direction. Migration increases genetic variation within populations and decreases divergence between populations, whereas genetic drift decreases genetic variation within populations and increases divergence among populations. Mutation increases both variation within populations and divergence between populations. Natural selection can either increase or decrease variation within populations, and it can increase or decrease divergence between populations. It is important to keep in mind that real populations are simultaneously affected by many evolutionary forces. Evolutionary forces which preserve genetic variation. Selection generally leads to decrease in variations, but the process which preserve genetic variations are 1. Balancing selection. Natural selection maintains 2 or more forms in a population. 2. Heterozygote advantage. Heterozygotes are more fit than the homozygotes. 3. Negative frequency-dependent selection. The fitness of a phenotype declines if it becomes too common in the population. 4. Neutral variation. Many of the nucleotide differences in non-coding sequences appear to confer no selection advantage or disadvantage.